Next up, we have Associate Professor Angela Chou. Angela completed a PhD in her postdoctoral fellowship at the Garden and joins the colleague as a cancer researcher at the end of this year, the start of this year, and she was recently named, as you heard last week, as the Outstanding Cancer Research Fellow of the Year by CNSW, looking at her research in pancreatic cancer. So thanks for sharing with us.
So first of all, I just want to thank Sydney Vital for the funding to do this research. So I represent the Cancer Diagnostic and Research Group here at Full Institute, which is headed by Anthony Gill. And so our group is really mainly focused on kind of introducing and translating kind of diagnostic tech to clinical, into the clinical lab, especially the pathology lab, and so that we could have the immediate impact on clinical care. And so this particular project which we looked at, which has now been published, named NTRK gene rearrangements, are highly enriched in MLH1, PMS2 deficient, BRAF wild-type colorectal cancer carcinoma. The kind of goal of this specific project is really to kind of look at the incidence of NTRK rearrangement colorectal cancer, and to see if there's a kind of a screening method which we can introduce into routine diagnostic care to kind of T-cell diagnose these cancers. And the reason for that is that, which I'll go into a bit later, is that there's a new tyrosine kinase inhibitors that target these specific gene rearrangements. So just as an introduction, colorectal carcinoma is the third most common cancer. So data from 2020 predicted for this year to be about a bit more than 15,000 cases which will be diagnosed in Australia. It's also the second most common cause of cancer death in Australia, predicted to be over 5,000 cases in Australia this year. So how do we treat colorectal cancer? So for localized disease, which still maintained within the colon, the first line of would be surgery plus or minus neoadjuvant and adjuvant therapy. In about 20% of the cases, patient will be diagnosed with metastatic colorectal cancer either at first presentation or later development metastasis. In routine pathology practice, we would do a KRAS mutation testing to see what treatment line they should go to. So for those cancers in 50% of cases which are driven by a KRAS mutation, these patients will have first line usually a 5-FU-based chemotherapy. And then in the other half of the cases which are KRAS wild type, so no known cancer driver, these patients will have an EGFR targeted monotherapy, or with chemotherapy, but not all of these patients respond well to this treatment. And so this is kind of KRAS wild type colorectal is a group that we're kind of quite interested in, and more and more data has shown that there's a group of proteins which are the family of tyrosine kinases probably involved as a driver in colorectal cancer, and hence the reason we're looking into it. So like I said, in the last 10-20 years, many of these tyrosine kinases have been shown to be very important in driving cancers. And because these a lot of drug companies have made a specific monoclonal antibodies to target specific gene aberration involving these tyrosine kinases, and the best example will be like HER2 therapy, anti-HER2 therapy for targeting HER2 amplification in breast cancer and gastric cancer. And then also in lung cancer would be anti-EGFR therapy targeting specific exon 19 deletion or mutation involving the EGFR genes. And also, like I said, these tyrosine kinases can also be aberrant from gene rearrangements, and these are also very important. So an example of this will be crizotinib in lung cancer for alpha rearrangement ROS1 lung cancer. So these tyrosine kinases have mainly been more kind of specific for a cancer type. But more recently, like I mentioned, so they're drugs which are more developed as a tissue agnostic drug. So they are designed to be used for any kind of solid cancers which have that specific gene aberration. So late in the kind of end of 2018 and 2019, two new TRK inhibitors which target there's 3 genes which are involved. So NTRK1, 2, and 3, which targets solid tumor approval by the FDA to target solid tumors which have these gene rearrangements. So, as I said, there's some recent data showing these NTRK rearrangement also occur in colorectal cancer, and they're mutually exclusive to the KRAS and BRAF mutation. So these currently diagnostically, there will be no way that these rearrangements will be detected in routine practice. And I think part of the reason is because in Australia, patients with metastatic cancer are not kind of routinely submitted for multi-gene molecular testing. If they can afford it, we'll send it to, for example, Foundation One. And I guess part of the problem is that these kind of rearrangement incidence are low in colorectal cancer, so there's an issue with whether it's cost effective. And so, like I said, hence for this study, if we could actually use a screening method by detecting the protein expression, then it would be really ideal. So, for our project, in our group, we have 4,569 very well annotated colorectal carcinoma samples. These have really detailed clinical data, including outcomes. Also pathological data, including the mismatch repair status and the BRAF mutation status. And so previously we have looked at 2 of these gene tyrosine kinase rearrangement, ALK and ROS1, and these appear to occur at very low incidence. In fact, there was only one case that had a ROS1 rearrangement in our study. And so we really want to see with this new drug approval whether what's the frequency of NTRK1, 2, 3 fusion or rearrangement in colorectal cancer, and see if we can use overexpression of the TRK protein as a screening method. So for the pan-TRK overexpression, we use the immunohistochemistry method on FFT material, and we use 2 different monoclonal antibodies to compare any differences. And you see on the right panel the picture. So depending on the type of fusion and the partner that's present, there's some difference in staining pattern. But most of these the positive cases, they also all show cytoplastic staining. And so to confirm that the rearrangements are present, we did RNA-seq testing, and also ultimately we also use a fluorescent in-situ hybridization using the NTRK 1, 2, 3 probe, just to kind of double confirm the findings. So result from this study, what we found was that in the 4,000 plus cases, there was diffuse high TRK expression in about 0.2% of cases. And these were all confirmed using RNA-seq or FISH testing to have either NTRK1, which is most common, or NTRK2 fusion. And also the partners, except for one, which didn't have enough RNA present, was confirmed at FISH, and we were able to confirm the fusion partner, but not shown here. And also when we compare the data with the mismatch repair status and also the BRAF mutation status, we found these cases were highly enriched in the cohort, which was mismatch repaired and also BRAF not mutated. So this is quite handy because as a screening method, because routinely we already test for mismatch repair as part of the Lynch syndrome screening program, and we also routinely test for BRAF screen for BRAF mutation using a immunohistochemistry method. So we picked up these cases which have this kind of phenotype, and then do the NTRK testing, then it will be enriched to 5.3%. Also, this group tumour appears to be more commonly present on the right side, and they also are large in size. I'm not shown here. There appears to be a trend for them to behave a bit more aggressively, but it didn't statistically did not reach a significant p-value. So, in terms of the impact of this study, like I mentioned, we actually do this routinely in our lab now. So a patient with metastatic colorectal cancer, so as part of routine, the MSI and BRAF status would be performed at the time of diagnosis. And like I said, if they were MLH1 or PNS pure, they had mismatch repair deficiency, and they're also wild type for BRAF 600E mutation. You can then do the pan-track immunohistochemistry to screen for a high expression, and if positive, you can then confirm that molecular testing and hence cost for molecular testing. And that's it. Thank you.